So in this review video, we're going to focus most of our time on G-protein coupled receptors, which are insanely fascinating to discuss. And at the very end, we'll talk a little bit about enzyme coupled receptors for a little while. We'll focus on RTKs, or receptor tyrosine kinases, but we won't spend too much time on those. They're far more simple. So for G-protein coupled receptors, they are a 7-trans membrane domain protein. So that means if this is the receptor head, which is going to bind to the ligand, there are 1, 2, 3, 4, 5, 6, 7 different membrane domains that are hydrophobic that go through the cell membrane. And to take a quick cumulative moment here, especially since the cumulative final is beginning to loom just a bit, this receptor of the G-protein coupled receptor system is a protein. It has a gene that encodes it. That gene is transcribed and translated. Upon the early stages of translation of this gene, the messenger RNA that encodes it, there is a signal sequence that is recognized by an SRP. And that SRP brings the signal sequence to an SRP receptor, which then threads it to a protein translocation channel. And finally, we have many start and stop transfer sequences. A stop, a stop, a stop, a stop, a start, a start, a start, that thread this protein through the membrane. This receptor was then loaded into a vesicle that was clathrin coated and carted off to the cell membrane, where it became part of the cell membrane protein system. And we can back up a little bit more and say that the gene for this receptor protein has a promoter that was bound by the TATA binding protein as part of TF2D. That reassembled the PIC, and ultimately that gene was transcribed by RNA polymerase II. The messenger RNA was processed by getting a cap, a tail, its intron spliced out, and it's circularized, and then off to the cytoplasm it went for translation. So at this point in the course, you want to start getting that kind of holistic, integrative, synergistic, cumulative way of thinking, tying all of these stories together. But here is our receptor. I've only drawn the receptor, but this is a G-protein coupled receptor, so let's draw in the G protein here. The G protein is made up of 3 domains. There's an alpha domain, a beta domain, and a gamma domain. And now we have a G protein coupled receptor. Again, to drive the point home, this is the receptor, this is the G protein, and together it is a G protein coupled receptor. So this is what's going to transduce the signal from outside the cell into the cell proper through signal transduction. So the first thing that happens is we do receive a signal. So out there in the external environment of the cell, there's a signaling molecule being released. This could be a hormone, this could be interleukin, this could be a small peptide, this could be any kind of signal molecule that's released in the external environment. And it is the primary messenger. It is carrying information. Now that information needs to be received by the cell, and the punchline of this story is that there needs to be a change in cellular behavior in response to that signal. That's really the grand goal here. This signal is saying, hey, there's too much glucose in the blood. Hey, there's a tiger outside and we need to mobilize energy stores because we got to climb that tree in a hurry. Hey, I smell fresh baked bread. Let's get the salvation response going because we're about to eat breakfast. This signaling molecule is carrying information. And that information needs to lead to a change in cellular behavior that takes glucose in from the blood, that mobilizes ATP stores for climbing that tree or fighting that tiger, that begins salivating in preparation of digestion of that fresh-baked bread. We need a change in cellular behavior in response to this information we are receiving. So the first step is that the signal molecule, the primary messenger, binds to the receptor. And in binding to the receptor, it initiates a shape change, a wave of motion that causes the receptor to change its conformational shape. For those of you that have taken biochemistry already, this will be very familiar to you. For others, just know that since structure equals function with proteins, every time a protein modifies its shape, it modifies its function. And so we have this kind of cascade, this wave of shape change down the receptor. What that shape change ultimately does is it jostles or disrupts the G protein. And the G protein reacts in kind. In fact, what the G protein does in response to this shape change is that a guanosine diphosphate is released by the alpha subunit, and a guanosine triphosphate is taken up instead. And I'll show that by filling in this G protein, this alpha subunit of the G protein, but with green. So now it's bound to GTP. And this G protein is now on. It is now active in response to having GTP. And so it leaves its two subunits, and it migrates along the inner surface of the membrane to another membrane-bound protein, an enzyme, in fact, that's called adenylate cyclase. And this active G protein activates or turns adenylate cyclase on. Now, what does adenylate cyclase do? It might not surprise you to hear that adenylate cyclase cyclizes adenylates. And it makes cyclic adenylate monophosphate, or adenine monophosphate. It makes cyclic AMP. And the precursor for that cyclic AMP is ATP. So adenylate cyclase takes ATP and turns it into cyclic AMP. Why did it do that? Well, it did that because this active G protein is sitting on it. Why is that G protein active? Because it's bound to GTP. Why is that G protein bound to GTP? Because the receptor jostled it when the receptor was bound by the primary messenger. So all of this is happening right now because the primary messenger bound to the receptor. That cyclic AMP, once it is made in the cell, goes and activates. So here's cyclic AMP as a black dot. It binds directly to and activates a protein called protein kinase A. And what do we know about kinases from this lecture and the previous one? We know that kinases change cellular behavior. Kinases phosphorylate things. And by adding phosphate groups to target proteins, they turn some proteins on, they turn other proteins off, they drive the expression of new genes, so new proteins are made in the cell. A whole array and suite of proteins is acting differently now because of protein kinase A being active, and that means we have a change in cellular behavior. The cell is responding to the signal. Now, what we also have to appreciate is that this alpha subunit of the G protein is also a GTP ACE. And we've heard about GTP ACEs before when we talked about tubulin. That means it can cleave the GTP that it's holding on to and cleave it to GDP. When it does that, this alpha subunit is now bound to GDP. That's the inactive form. It will migrate back to its original position and leave adenylate cyclase. That means adenylate cyclase will no longer be on, cyclic AMP will no longer be made, protein kinase A will no longer be active, and the change in cellular behavior will cease. If the signaling protein is no longer present on the receptor, then this is a good thing. The glucose is out of the blood, the tiger has moved on, the fresh baked bread is gone, and we no longer need this cellular response. We no longer need this change in cellular behavior. So it's good for the G protein to kind of go back to the receptor and say, hey, is that message still accurate? Is that message still relevant? Is that actually still going on? And if the answer is no, then the change in cellular behavior should stop. But if there is still glucose in the blood, if the tiger is still looking at you, if there's still fresh baked bread around, then as soon as this alpha subunit goes back to the G protein, it's going to get hit with that conformational change again. It's going to get let go of the GDP that it just made. It's going to pick up a new GTP, and then you know it's going to head right back to adenylate cyclase. It's going to turn adenylate cyclase on. Adenylate cyclase is going to go back to making cyclic AMP out of ATP as its precursor, and that cyclic AMP is going to turn on pKa, and the change in cellular behavior will persist. A really, really nice system where the G protein goes back to the receptor every so often, as far as its GTP ACE activity allows it, and checks in. Hey, is the primary messenger still there? No? Then I'll sit here dormant and wait. The cellular behavior goes back to normal. Hey, is that primary messenger still there? It is. That message is still relevant. Well, let me drop my GDP, pick up a GTP, and then back off to adenylate cyclase I go to keep that change in cellular behavior going. So that's the cyclic AMP as a second messenger story. Now we're going to tell a second story of G protein coupled receptors, and it's going to have some of the same hallmark features here. So one of the things that it's going to have in common is that we will end with the same punchline, a change in cellular behavior. And the other thing that it's going to have in common is a G protein coupled receptor system. So that's going to also be something that we can leverage. But this story doesn't use cyclic AMP and it doesn't use adenylate cyclase. It's a little bit more complicated and has a few more steps. But again, we will leverage what we already understand and we'll know where we're heading, a change in cellular behavior, and we'll know how we're going to get there through an active G protein. So the ligand has bound. This primary messenger is present on the receptor. We get that wave of motion, and we're going to let go of our GDP, and we're going to take up a new GTP. We're going to activate our alpha subunit, and our alpha subunit is going to translocate along the membrane, and it's going to bind to, it's going to bind to a membrane-bound enzyme, just as we saw before, and it's going to catalyze some changes. Now, the membrane-bound protein or enzyme that we are bound to now is not adenylate cyclase. It's an enzyme called phospholipase C. Now, sucrase cuts sucrose, galactase cuts galactose, lactase cuts lactose. What do you think lipases cut? Lipases cut lipids. So this is phospholipase C. Well, phospholipids, we know, are membrane components. So what this enzyme is actually going to cut is a membrane lipid, and it's going to cut it roughly in half. So when phospholipase C is active, why is it active? Because there's a G protein there. Why is there a G protein there? Because it's bound to GTP. Why is that G protein bound to GTP? Because we have a primary messenger on the receptor. When phospholipase is active for this reason, it will cut a membrane lipid. The membrane lipid that it cuts is going to be referred to as PIP2. That's an acronym for a much longer phospholipid name. And PIP2, when cut, is going to become two different halves, two different pieces. One small piece is called DAG, or diacylglycerol. That stays in as part of the membrane. The other half of PIP2 that's severed off is IP3. That's the other piece of IP3. So what do you get when you take IP3 and you covalently glue it to DAG? You get PIP2, this membrane lipid. Why was PIP2 cut into two halves, each half called DAG and IP3 respectively? Because phospholipase C was active, because the G protein is there, because the G protein is bound to GTP, because the GTP is the active form, because the receptor was bound by the primary messenger. So all of this is happening now because of that primary messenger and the action of the G protein. IP3 is going to go to the smooth endoplasmic reticulum, the smooth ER, running out of colors. And it is going to cause calcium channels in the smooth ER to open. So here's IP3. It binds to these calcium channels, and as a result, calcium comes rushing out of the smooth ER. IP3 has opened the smooth ER so that calcium can leave it and enter the cytoplasm. What does DAG do? Well, DAG, I'm running out of room. So I just shifted everything over to the left a little bit there just to give me more room. DAG stays as part of the membrane, but it activates and turns on an enzyme that is also in the membrane called protein kinase C. Not protein kinase A, that's from the previous story, protein kinase C. And protein kinase C is going to phosphorylate and therefore open calcium channels in the cell membrane. So now calcium comes rushing in from outside the cell as well. So we've got calcium rushing into the cytoplasm from the smooth ER, and we've got calcium rushing into the cell from the external environment, from the extracellular environment. So before we go any further on the signal cascade, let's kind of back it up a little bit. What we have at this point is a lot of calcium in the cell. And we're still not quite sure how we're going to get that change in cellular behavior that we know we need to end on, but we do know that we have lots of calcium in the cell. Why do we have lots of calcium in the cell? Because calcium came rushing into the cytoplasm from the smooth ER stores. IP3 caused that to happen. Why do we have a lot of calcium in the cell? Because calcium came rushing in from the external environment through calcium channels at the cell membrane. Why did that happen? Because protein kinase C phosphorylated and opened those calcium channels. Protein kinase C was activated by DAG, the other half of PIP2. So DAG indirectly caused calcium to come in from outside the cell. IP3 directly caused calcium to come into the cell from the smooth ER, and both DAG and IP3 are byproducts of PIP2 cleavage. Phospholipase C is what cleaved PIP2. Phospholipase C is active because of the G protein, and the G protein is there because of the presence of the primary messenger. Now, this calcium interacts with a protein that is modulated by calcium levels. So calcium, when it's present, will turn this protein on. And since this protein is modulated by calcium, its name is calmodulin. Calmodulin, when it is on, will activate an enzyme called calcium modulated kinase. Oh, here come the goosebumps, because finally, we now have the opportunity for a change in cellular behavior. This calcium modulated kinase will then go and phosphorylate a whole suite of target proteins and turn them on, turn them off, drive their expression, and result in a change in cellular behavior. So let's take a brief moment here to go through it again. A primary messenger binds to a receptor, a 7-transmembrane receptor at the cell membrane, and initiates a wave of motion. That wave of motion causes the alpha subunit of the G protein to let go of its inactive GDP and pick up GTP. That turns the G protein on. It migrates to phospholipase C, which as an enzyme cleaves PIP2, the membrane component, into two halves, IP3 and DAG. DAG migrates over to and activates protein kinase C, and protein kinase C, as a result of that, phosphorylates calcium channels in the cell membrane, allowing calcium to come rushing in from outside. IP3 migrates over to the smooth ER, where it directly causes calcium channels in the smooth ER to open so that calcium can come rushing into the cytoplasm from intracellular stores. The net result of all that is a huge increase in calcium levels. That calcium ion will bind to the protein calmodulin, whose activity is modulated by calcium. In the presence of calcium, calmodulin is on. Active calmodulin then activates cAMP kinase, or calcium modulated kinase, and active cAMP kinase will then phosphorylate a huge array of proteins, resulting in a net change in cellular behavior. In all this time, along the way, the alpha subunit G protein has cleaved its GTP back to GDP. And as a result of that, it will leave phospholipase C and go back to the G protein coupled receptor. Now, if the primary messenger is still there, that G protein will take up a new GTP and go back and catalyze all of this again. But if instead that primary messenger, that ligand, is not in the receptor, phospholipase C will cease being active. PIP2 will no longer be cleaved. Phospholipase C. We will no longer have DAG made. We will no longer have IP3 made. In the absence of DAG, protein kinase C will not be active. we will lose the phosphorylation of the calcium channel. Those channels will close, and calcium will no longer come in from outside the cell in the absence of DAG. In the absence of IP3, calcium channels in the smooth ER will close, and calcium will no longer come into the cytoplasm from the smooth ER. Calcium levels will drop in the cell as a result. Without calcium present in the cell, calmodulin will be off, not on. It will be unable to activate cAMP kinase. Inactive cAMP kinase will not be phosphorylating its suite of target proteins, and the change in cellular behavior will cease exactly as it should be, because there is no longer a primary messenger at the receptor. That is the calcium as a second messenger story. A lot more complexity, a lot more moving parts, but the theme of it is the same as the cyclic AMP story. The binding of a primary messenger to a G protein-coupled receptor changes membrane-bound enzyme activity, resulting in the release or production of a second messenger, cyclic AMP in the first story, calcium here, and the net result of that is an active kinase that changes cellular behavior. To wrap this all up in a neat bow, let's talk about enzyme-coupled receptors. We'll talk about receptor tyrosine kinases specifically, but basically we'll tell the story of most enzyme-coupled receptors. So what makes enzyme-coupled receptors unique is that they exist in receptor halves. So this is the inactive form of the enzyme-coupled receptor, and the two halves of the receptor are separate and distinct, floating in the membrane, diffusing laterally. When the ligand for this is bound by one receptor half, it attracts and recruits the other receptor half as well. And so the ligand itself acts as a bridge joining the receptor halves together. It's the ligand binding that holds the two receptor halves together. Now, these are enzyme-coupled receptors, meaning that the enzyme is part of the receptor itself. This half of the enzyme-coupled receptor phosphorylates the other half. And vice versa, the other half phosphorylates the first. Now, technically, this is referred to as autophosphorylation or self-phosphorylation, but I want you to appreciate the fact that it's not really genuine autophosphorylation because these are two different subunits of the receptor, and they phosphorylate each other. One half phosphorylates the second half phosphorylates the first. Now, this is an active enzyme-coupled receptor in its phosphorylated form. It can either phosphorylate anything that comes near it, so any target protein that comes in the vicinity of the receptor can be phosphorylated itself. That happens. It's not all that common, but that can happen. Or more commonly, we have new enzymes that are recruited to this phosphorylated form of the receptor, and these new enzymes then become active, and they may float off as kinases in their own right and phosphorylate target proteins in the cell. That's more common and happens more readily. That's basically all we need to say about enzyme-coupled receptors in general. Receptor tyrosine kinases follow this story. They have that name because the residues that are phosphorylated here in the receptor are tyrosines. That's the target residue that's phosphorylated by these enzymes. They're just a specific class of enzyme-coupled receptors. Some things to note here, though, is that the receptor itself is the enzyme. It is the kinase that is responsible for turning on other kinases to achieve a change in cellular behavior. And this notion of the ligand bridging the two halves of the receptor together without the ligand. If we take the ligand away, then these two receptor halves will float apart. Since they require one another to phosphorylate each other, as they float apart and those phosphates are removed, they're back in their inactive form, and they cannot autophosphorylate. I hate that term because it's not really true, but they can't phosphorylate each other until the next ligand comes and serves as the new bridge, bringing them together. So once again, they can phosphorylate one another and reinitiate this change in cellular behavior. That's really all we need to say about enzyme-coupled receptors or receptor tyrosine kinases specifically. There are some implications between G-protein coupled receptors and receptor tyrosine kinases with regards to their potential for amplification. Amplification is another really big part of the signal transduction story, but we'll leave that for a challenge question that's coming up a little later in the week.